Hey, Sterling here, Vagus Nerve Stimulation and Repair. We're going to be talking about how does a nerve get affected and how does a nerve regrow after being treated with ultrasound. So we're going to go through some of the proposed mechanisms of action for how a nerve is actually able to repair itself after being stimulated with ultrasound. So we're going to start here with this really cool paper. That's a case study of an actual amputee regaining sensation and muscle function in a residual limb after peripheral nerve stimulation with intense focused ultrasound. So this is focusing on intense focused ultrasound, but it doesn't just have to be the intense ultrasound. This can also apply in other cases, as has been shown, to normal, low intensity, low pressure focused pulsed ultrasound as well, or diffuse ultrasound. So this is really cool. This study conceptually, which I think is really huge, is that the study conceptually overlaps with others that show ultrasound stimulation of the thalamus can reanimate neural circuits. So this has been shown. This is something that specifically works, and this actually happened after a year of nerve function dormancy. This is really huge. So here's a picture of what this looks like specifically. I'll blow this picture up a little bit. And where this is just under the skin. There's a tibial nerve, and they stimulate it with ultrasound. This is a standard, number 4, standard ultrasound scanner, a linear ultrasound scanner operating in conjunction with a large transducer that's designed to focus ultrasound on one area to specifically target that tibial nerve with a high area of impact. So additionally, yeah, this is just below the knee actually. So yeah, so they stimulated with the ultrasound and it regained nerve function was regained after stimulating a peripheral nerve with ultrasound. So how does something like this work? You're like, what's going on here? So what I wanted to have us do is look at the dynamics of an actual nerve. So we can see the nerve body. So this is a typical neuron. We're looking at more of a central nervous system style of nerve where there's a nucleus, cell body, there's axons, there's dendrites, there's receptors, there's myelin sheath. Now, what's not shown here is that if you go deeper into the axon, then what you're gonna start to see is the cytoskeletal tissue. If you look really down here at the axon, you're gonna see that the cross section has been cut. Now, if you did a cross section analysis of that specific axon, you'd probably find there anywhere between 20 to 100 microtubule cytoskeletal structures that span all the way from the cell body all the way down to the axon terminals. So if we look here, this is a kind of a side look at the inside, on the right side, the inside of a dendrite, and this is a dendritic to axon connection. So axon, dendrite. This is the axon, which we see. So if we see axon terminal connected to what would look like the dendrites of an adjacent neuron just below this. So we had 2 of these stacked on top of each other. We would see axon coming down, connecting with the dendrite to connect with the rest of the cell. So we have axon on the left side with these microtubules. These are like the zebra striped cross sections, sending out signals, carrier signals to the synapse, receiving those signals, and being received at the level of the microtubule in the dendritic spine, or inside of the dendrite actually. So microtubules are a proposed mechanism for neural functionality actually. So if we look at what a microtubule looks like on closer inspection, it's a tubulin tube basically. So if you've seen let's see, carbon nanotubes, you will be familiar with what that looks like. So a carbon nanotube is a small tube consisting of repeating sections of carbon, benzene rings essentially, just stacked back to back to back, wrapped in a circle. So a microtubule, similar type of core building block. Like, I think of them as like tubulin as an IKEA building block. You just stack them to create as long of a structure as you want. They polymerize and then they depolymerize. When they polymerize, they zip up like a zipper. They create a flat sheet and then they zip up like this. And then when they depolymerize, when they fall apart, they split apart like a banana peel. Okay, and they get capped. A cap goes on the very top to prevent the depolymerization. So cells that exist for a lot longer, like the ones in your brain, usually have caps on them that prevent depolymerization, so they're more stable. And the ones that grow in organisms that are expanding rapidly and shrinking, or cellular reproduction in your skin cells, you'll see a lot of microtubules are polymerizing to grow to create the meiosis and splitting of the cell, and then depolymerizing once their use has been expended. And this process goes on and on and on. So in the case of a non-functioning nerve, due to damage or chemical damage or physical damage, what you would pretty much expect to see is a lot of depolymerization of these microtubules, because again, unless a microtubule has been capped, it's unstable. So if you cut it right in the middle, it's going to depolymerize right through the very middle, and that's going to cause depolymerization and dysfunction of the axon and the dendritic connections. So nerves don't work, muscles don't work, cells aren't communicating, neurons aren't working. Your central nervous system can have this. This is effective neurodegeneration. Alzheimer's, Parkinson's, MS will typically see this depolymerization across the board. Loss of memory, loss of function, loss of interconnectivity, decreased alpha signature. If you did an EEG scan of the human brain, you would see all these signatures of what would appear to be, on the surface, depolymerization of mass amounts of microtubules. Depolymerization is how you lower your brain function, your central nervous system function, and neuronal function. Polymerization and capping is how you increase the connectivity between your brain and your body, and that's good for everybody. But you can have too much, but that's a whole different video. Now, if you look at the microtubule on even closer, we can see the cross-section. There are 13 rows around. They're stacked with tubulin dimers. There's an alpha and a beta tubulin dimer, and they all stack just like this. Inside of them, there are contained many different amino acid proteins, building blocks of a tubulin. Each of them have specific binding sites for different kinds of proteins, different chemicals, different carrier signals, even potentially electrical signals that can travel through the edge of these microtubules in these lattice electrical magnetic patterns that some have proposed, like Stuart Hamroff, MD, at University of Arizona Tucson. He's an anesthesiologist who pioneered a lot of this research. Hypothesized that microtubules have calculation potential inside of them, so they can basically work like an abacus, essentially. So your brain and your body have microtubules, which were considered to be dumb building blocks that just provided structure. Now we've found that they might actually have some ability to calculate things in real time, meaning you have distributed computers all over your entire body, mostly through your entire central nervous system, in your spine, your brain, pyramidal cells in your fourth layer of your cortex. And I definitely hypothesize that your vagus nerve specifically contains a lot of these microtubules in such conditions where they're protected from extraneous external effects, giving them the ability to contain the same processing features that your brain may have. Sort of like the vagus nerve being the irrigation, or the irrigation system of your body through microtubule interconnection. So the health of your microtubules is the health of your vagus nerve, is the health of your brain, is the health of your entire life. So your microtubule function is really, really crucial. Very, very important to have good, healthy microtubules. So looking back, if you look at this case study again, we look at the features of ultrasound to stimulate microtubules, specifically to stimulate microtubule outgrowth. There's some research that's very compelling that shows that specific pulses of ultrasound can spontaneously create the creation of microtubule polymerization, spontaneously. So anyway, that's it for this video. Just wanted to do a fun little deep dive into what a proposed mechanism of action is for why ultrasound is such an effective tool for repairing nerves, for increasing neural function, increasing brain density. There's a lot of stuff that ultrasound on the human brain and body can do, specifically the vagus nerve. In this case, in the cervical vagus nerve domain, it works incredibly well, and you see results really, really fast. So anyway, that's it for today. Thanks so much for watching. If you want to see more videos like this, make sure you subscribe. Make sure you turn the bell on. We'll be producing some pretty incredible content because the world's got to know about this stuff. All right? Thanks so much. Sterling from Vagus Nerve Stimulation and Repair. If you want to go join us on our Facebook group, the link will be in the description. Thanks so much. Take care. Bye-bye.